Hi everyone. So today I'm going to talk about the evolution of the accessory genome. And I'm Raymond Chen. I'm currently a research associate at Imperial College London. So to start with, I would like to invite everyone to think about this question. Why is diversity so persistent? We can think about examples in ecosystem, human behaviors, and bacterial population. Why we see so much diversity there and why it persists. So for today's outline, I will cover some topic about pangenome and what are core genes, what are accessory genes. I'll talk about negative frequency dependent selection and also how it is used in microbial genomics and how can we predict the post-vaccine era evolution. And there are like several remaining questions. But actually, the only thing I would like to talk about is about negative frequency dependent selection. I will give you I hope to after today you will have some concept about this selection. So first of all, let's just try to define what is pangenome. So pangenome is the collection of all of the genes within a species, and we can further um, divide the pangenome into core genes and accessory genes. So uh, if you see this figure, you can see like in the middle where every isolate overlaps to each other. These are the core genes that are carried by almost all the isolates within a species. And other parts are the accessory genes, which only carried by several isolates or one or two isolates. We can also look into another way. We can look into the specific clade in one species and also a fourth species tree. When we look at the core genes, you will see the number of core genes doesn't change a lot when we look into clade, a specific clade or in a full species tree. But for the accessory genes, you will see in a specific clade, there are just a few genes in a specific clade. But when we look into the full species tree, it's changed a lot. The number of accessory genes increase a lot. So here comes to an obvious question. Why do we have so many accessory genes which are in at their intermediate frequencies? What is the force that maintain these phenomena? So because if the genes are beneficial to the isolate or the species, then it will be fixed. It will increase its frequency and it will be fixed in the population. If the genes are detrimental for the population, then it will be eliminated. So why do we see so many intermediate frequency genes? There are like several theories to explain the observed diversity. So first of all, the neutral theory. It talks about the random fluctuation throughout the evolutionary process. So the fact with the observed diversity we see is just like from the randomness throughout the time. And also there are heterozygotes advantage. So in some scenario, uh, having having heterozygote genotype actually having a best fitness in the population. So it will remain the diversity. And there's also another force, which is negative frequency dependent selection that try to maintain the diversity. So what actually NFDF is? So by definition, NFDS describe the fact that the fitness of a type decreases as the frequency increases. So if you look at this figure, in this environment, there are two types. One is purple type and one is the blue type. So, and they exist in the same environment. And in both two types, the x-axis is the relative abundance and the y-axis is shows the fitness of the two types. So in both two types, you can see as the frequency increase, the fitness decreases. This is by the definition, the NFDS selection. You can also flip the purple type horizontally. So here for the purple type, the x-axis still shows the relative abundance, but the leftmost one is the highest frequency and toward the right-hand side, it's decreased the frequency. So if you see the left-hand side scenario, this indicates the fact that the population is full of the purple type and without any blue types. But once the blue type invade in this purple type population, it will start increase its frequency because the blue type fitness is higher than the purple type. And it will increase until like this point, the point 40% blue type and 60% purple type. It will reach this equilibrium. You can do it another way around with the purple type invade into the blue type, and you will reach the similar equilibrium. So this dynamic shows how an NFDS to maintain the diversity. And there lies a lot of intuitive examples of NFDS in our life. So there's a very classic example about the orchid color polymorphism. So in this example, in this specific orchid species, there are two flower types. One is purple, one is yellow, and both of them are not really tasty for the pollinator. So if the purple type is very common, then the pollinator will learn that purple type is not tasty, so they will turn to the yellow type. And then so the yellow type will increase its frequency. But once the yellow type becomes too common, then the pollinator will learn that, oh, the yellow type is not tasty. So they will turn back to the purple type, and the cycle will continue. There's also some cultural chain that have this NFDS dynamic. So because like we can think about like art, fashion, these kind of thing can fluctuate around. So one example is about people's name. So people will usually think that our name is not cool if too many people share the same name. So you can see like people want to be cool. So once like William become too common, then the popularity of William will decrease. Then there will be more people with rare name increase. So this is an example about how cultural trend is maintained by the NFDS. There's also example in sport area, like I'm personally a very crazy baseball fan, and there's a big thing in baseball that the left-handed pitcher is better to deal with the left-handed batters. They always combat the left-handed batter. So if you know your opponents have lots of left-handed pitcher, then you will equip your team, with lots of right-handed batter. And in response, your opponent will equip more right-handed pitcher. So still, this cycle will continue. Yeah, so there are like so many examples about NFDS, but how can we actually prove NFDS exists? So experimentally, we can do this kind of experiment, which is called reciprocal invasion experiments. So this is using yeast as an example. So for yeast, there are several strategy to utilize nutrition, and in this case, there are like galactose on and galactose off strategy. So the yellow one is showing the galactose on strategy. So once if the environment is rich in nutrition resource, which means that you can use both glucose and galactose, then it will maintain this NFDS dynamic. So for example, if the population is full of the galactose on type, once you invade a galactose off type, then the galactose off type will increase its frequency. And you can do another way around. You can see the at the end it will reach an equilibrium frequency with both galactose on and galactose off type exist together. So yeah, this is one of the experiments which you can demonstrate there is mutual invisibility, and that is actually a strong proof of NFDS. So back to the theories. uh, I know people come here want to know about bacterial population, not about human behavior or yeast. So, how can we use this theory to explain the observed bacterial diversity? So, back to the neutral theory. Neutral theory, from the randomness, it can actually create some diversity, but this kind of diversity is transient. So, you can look at this figure. This is based on neutral theory. You can see the allele frequency can fluctuate between 0, and 1, but at the end, in the long run, it will either be fixed to 1 or it will be eliminated. So the diversity created by neutral theory is a transient phenomena. There are also different theory that can maintain the diversity in the long run. So one example is about heterozygosity advantage. One classic example is the human sickle cell anemia. So because under the malaria condition, the heterozygous with sickle cell anemia will actually survive better in that kind of condition. So having heterozygous will give individuals at more advantage to survive. But actually in bacteria, as you will know, bacteria are haploid and sorry are haploid. So this kind of heterozygosity advantage is not really common, but there are some examples in plasmid and duplicated genes. So here comes the other theory. So the NFDS is also another way that can maintain the long-term bacterial diversity. So here I'm going to give you an example about how NFDS maintains pneumococcal diversity, and this is based on this paper. This is a very fundamental paper to demonstrate how NFDS can maintain the bacterial diversity. So in this study, they include three very diverse populations, one from Mela in Thailand, one from Nijmegen in the Netherlands, and one from Massachusetts in the US. So first of all, they look into the lineage frequency distribution in different populations. So for example, in this figure, they look at the lineage frequency in Massachusetts versus lineage frequency in Mela in Thailand, and also Massachusetts versus Nijmegen. And they find that there is not no much correlation between the populations at lineage level. But when they look at at gene level, there's actually a high correlation of gene frequency in different populations. And then when they look into because for the pneumococcus we have vaccine and the vaccine will target the certain vaccine type. So we wonder like how does the pre-vaccine population pneumococcal population correlates with the post-vaccine pneumococcal population. So you can see also here in at lineage level and at gene level. So this also demonstrates that at gene level, we can see a high correlation between the pre-vaccine bacterial population and post-vaccine bacterial population. So why do we see this phenomena? What is the force that maintain this phenomena? Surprisingly, they are using the multi-locus NFDS model. So this multi-locus NFDS model is a combination of the neutral model plus the the model that the MFDS model that will actually affect the offspring number. So let us consider about what will affect the offspring number. So there are majorly four components. First of all, the carrying capacity. So this is quite intuitive because we have limited resources. So we can only contain a limited bacteria population. And then there is migration. So when there is a migration event happen from A to B, then you will see the population in A will decrease, the population in B will increase. So you will see the change of offspring number. And then for the pneumococcus, there is a strong vaccine pressure. So you will see the vaccine type will be target and the vaccine type population will decrease. Uh, in contrast, the non-vaccine type bacteria will increase. And last but not least, there's NFDS effect on the pneumococcal population. And this is the most important thing I want to talk about. So if we put all these four components in the formula, so the observed number of offspring will be affected by these four components: carrying capacity, migration, vaccine pressure, and the strength of NFDS. And when we look more into the NFDS components, there's two parameters we are going to estimate. So the first component is the pi, which is which tries to explain the deviation to the previous equilibrium frequency. So in pneumococcus example, we assume that before the vaccine implemented, the pneumococcal population already reached equilibrium. So there's a pre-equilibrium frequency. And then we now collect data and we try to observe the current frequency of each gene. And here it's sum all of the gene all together. So it is a multi-locus NFDS model. It's consider multiple accessory genes all together. So for example, for the rare genes, in this case, let's assume the rare genes has the frequency of 0.1. Then if you do this math, you will see this will give you a positive number. And if this is positive and if the isolate carry a lot of rare genes, then the summation of this part will be a positive number. At the end, you will get a positive pi, and if you have positive pi here, it will increase the number of offspring at the end. In contrast, if this isolate carry a lot of common genes, so for example, the genes they carry are around 0.8, then you will have a negative number here. And at the end, you will get a negative pi, and then it will result in a decreasing offspring number. So this is how the deviation from the equilibrium will affect the offspring number. The other component in the NFDS part is the strength of NFDS. So we can classify the loci on the heterogeneous strength of NFDS by like just categorize like a proportion of genes that are under strong NFDS and a proportion of genes that are under weak NFDS. So by doing so, we can make a better estimate to say, oh, this set of genes that are under very strong selection pressure, and these sets are actually more neutral. And with this, we need to have another parameter which indicate the fraction under strong NFDS. So at the end, from this model, we would like to estimate the strong NFDS effect, the weak NFDS effect, and the fraction that is under strong NFDS, and also the vaccination effect, and also the migration effect. So I'm not going to show the model comparisons statistics, but at the end, in the result in the paper, the multi-locus NFDS model significantly outperforms the vaccine migration only model. And we can look into the results, and you can see it here in the Massachusetts population. The strength of vaccine is around 0.03, 0.04, but this is under the neutral model, and when we use the either homogeneous rate multilocus NFDS or heterogeneous rate multilocus NFDS, we will have higher vaccine effect. So why do we see this result? This result shows that there is an underestimation of vaccine effect using the neutral model. And this is because when the vaccine is implemented, the vaccine will remove the diversity. And back to the reason why we are talking about NFDS is that NFDS actually maintain and increases the diversity. So if we ignore the vaccine effect, sorry, if we ignore the NFDS effect, we will we will actually underestimate the vaccine effect so that we will see what we see, the observed diversity. But when we consider the NFDS effect, the genuine vaccine effect will be estimated. And another interesting point here is to show that the NFDS is not negligible. So this is not the zero. So this is quite a phenomena in the pneumococcal population. And you can also see this NFDS phenomena across different populations. So the biological implications here is that NFDS acts on the accessory genes, and gene but not and the NFDS is acting on genes but not strain level, and it will maintain an equilibrium for gene frequency. And actually, we can use this multi-locus NFDS model to try to predict the bacterial population after vaccine perturbation. So here comes a very direct application. With the model we have now, we can try to predict the post-vaccine pneumococcal population. So we can try to optimize our vaccine formula. So after vaccine implementation, the strains that with the strains survive are those strains that are non-vaccine type, and usually those non-vaccine type with carrying rare accessory genes will increase more than those carrying common genes. This is because of the NFDS. So different non-vaccine type do not increase equally. We need to have this model to try to predict more accurately how the non-vaccine type frequency increase. And with this, we can also consider the invasive disease risk and the AMR profile, the antimicrobial resistant profile. So we can actually try to optimize a vaccine that is actually going to decrease the disease burden rather than increase the disease burden. And an interesting point from this paper, it shows that increasing vaccine valency shows diminishing gain. So actually maybe increasing vaccine valency is not really a best strategy. We need to think more carefully about the NFDS dynamic. And also, there are like lots of bacteria that are very recombinogenic. So, pneumococcal is one example. So, we people will also wonder how the NFDS interact with the horizontal gene transfer or the accessory gene recombination. So this paper shows that the accessory gene recombination will prefer deletion over insertion. So there's a asymmetric rate. Usually, one isolate will have more deletion than gaining new gene elements from insertion. But if this is the common evolutionary force, a lot of accessory gene cannot survive because there are more deletion over insertion. So why can we still see so many accessory genes? And surprising or not, NFDS comes to rescue. So NFDS can prevent the decay of the accessory genome and maintain the multi-strains population. So now we are thinking about recombination and horizontal gene transfer. And NFDS tells us that each type can invade when they are rare. And NFDS actually acts on accessory genes. So let us think about migration event or horizontal gene transfer event into a new population. So horizontal gene transfer will allow introduced genes to be maintained in a new population because of the NFDS. So if we can try to quantify the speed, the rate that the gene transfer from one population to another population and combine it with NFDS, we can get a more accurate dynamic about how gene frequency fluctuate. And there's a framework that allows us to try to estimate the spread of the gene. So I have described like the phenomena on NFDS and how we can use the multilocus NFDS model to describe the observed diversity we have now. But why NFDS? Like what is the underlying reason that's give the selection force? So as you can see here, if we look into the accessory genes, the intermediate frequency genes, they are very enriched in several categories in bacterial genes, in mobile genetic elements and restriction modification systems, and also in surface protein. Um, if you stand on the bacterial point of view, bacteria are constantly facing a lot of difficulties. There are strain competition between strains, and they are also facing the threat from the phage. Phage are trying to invade the bacteria, and also they are constantly facing the threat from the host immunity. So because of these biological players, uh, there's NFDS to maintain the accessory gene diversity. So finally, I would like to sum up everything by how we can use NFDS to more applications. So first of all, we can try to understand the post-vaccination population shifts by the NFDS model, and then we can optimize the vaccine design to minimize the invasive disease burden. And then we can also try to describe why we see stable accessory gene frequencies across populations. And NFDS actually outperform the neutral models in forecasting strain composition. So back to the opening question, why is diversity so persistent? So if we think that this is the whole amount of observed diversity, there's a big chunk that is explained by the neutral model, the randomness throughout the time. But this diversity is transient, and there is another force maintained by NFDS that's actually maintain a long-term observed diversity. So here are some remaining questions I think we might need to investigate further. So first of all, how ubiquitous is NFDS? So recall that in the NFDS model, we can actually estimate what is the proportion is under strong NFDS, what is the proportion under weak NFDS. And I think we still need to have more study to understand what is actually the proportion. And then I think we can have more tests on to get more strong proof on NFDS other than reciprocal invasion, because not everything can be tested by doing this kind of experiments. And also, what are the ecological agents driving NFDS in bacteria? I've talked about phages, I've talked about immunity niche overlap, and there are lots of biological ecological players and we need more stories to describe this. By the way, I'm also working with an artist on the phage image, and so if you are interested in art, you can also look into the details here. And there are some other remaining questions about how does NFDS operating at one level, especially the gene level, interact with NFDS operating at different levels, like how it affects the species level. And at the end, how does horizontal gene transfer interact with NFDS? So with this, I think most of the work I've described today, it is mostly developed by Dr. Nick Croucher. So he developed the multi-locus NFDS model, and also there are lots of paper it's from him. So if you want to use the model, if you find some bug, please find him. And I think at the end, the NFDS model try to tell us like to be cool and to be unique, but don't be too popular. So thank you, everyone.
Thank you so much, Raymond, for the presentation. And thank you also for adding this little artistic touch at the end. So if you didn't manage to scan the QR code, you will have the slide tomorrow. So you will be able to scan and reach the page that it was linking to. So we already have a few questions in the Q&A. I will go in chronological order. So from the first one that was asked, which is actually quite precise. I think it's an acronym that you used in one of your slides. So one of the participants is asking, what is GCOG in your pneumococcus diversity slide?
Yeah, so there are lots of ways to define what is a gene is, and there's a different. I mean, how do we call the genetic variant? And GCOG is actually try to call the genetic variants by using like the presence absent matrix. So either you carry this kind of genetic variant or not. So actually in the model it's a 0, 1, 0, 1 binary matrix that. So that is just a way to call genetic variant. You can also use SNP. You can also use like KMers. And I think if you really want to use the model, those can fit into the model.
Thank you. If there are any follow-up questions, please feel free to keep on typing in the Q&A. So the next one is about the three theories that you mentioned since the very beginning of the presentation. So the neutral, heterogeneous, and NFDS. Are there studies which compare these three theories experimentally or theoretically?
So I mean, just from the paper I described about the pneumococcal population, it's actually comparing the neutral theory and the NFDS theory. So that is one of them. I mean there's a lot of them. And actually throughout the presentation I have put the citation there. I think many of them have some comparison between the neutral and the NFDS theory, because like neutral theory we always think this is a baseline we need to compare with.
Thank you. So they can follow some of the references that you have in your presentation, and this may help to provide further materials. Perfect. Thank you. So then the next question is always on NFDS. So if NFDS acts on genes and genotype with this gene is rare, what impact does it have on the genotype? So I think the question is on the general impact if one of the variants that has rarer frequency is pushed forward, let's say, by NFDS. What's the effect on the whole genotype for that given gene?
Yeah, I'm not quite understand, but I think that's like uh so once this gene is relatively rare, then it will give a positive fitness on these isolates. And if this isolate carry lots of rare genes, then overall it accumulates the effect, and it will give a more positive effect on the fitness.
So the next one is whether the NFDS model is limited to accessory genes or can also be applied to core genes in a population.
So I think, yeah, I didn't really talk about core gene today, but if you think about core genes, they are mostly carried by every isolate in a species. And why does it happen? It's because maybe they are functionally very important. And so usually this kind of gene will be more they will be have more purifying selection in core genes, because once a mutation happen, it is quite detrimental and it will be quickly wiped out in the population. So NFDS actually work on the accessory genes.
Thank you. The next question is on vaccines. So if vaccine valency is showing diminishing returns, what kind of new strategies do you think could break this pattern?
Uh, I think So because of the time, I didn't explain a lot on the vaccine valency thing. I think the important thing is when we increase, we can increase the vaccine valency, but we need to consider the dynamic after increasing the valency. So whether it will cause the rare genes, which might be very associated with invasive disease, whether those genes will increase. That will be an important thing. So it is not just about valency. It's about, first of all, we need to identify which are the causal genes that will affect the invasiveness of disease, and whether after vaccine implementation, whether these genes will increase their frequency. That is the important thing to think about. Yeah, there are lots of other strategies that we can target more conserve protein, or there are lots of discussion about vaccines. But I think just the most important point, we need to think about the NFDS dynamic.
Thank you. So the next one will be people use different terminologies to explain the pangenome. So what is the key difference between shared genes and accessory genes, or are both similar?
Yeah, so I'm not I so as you can see in my slide, I didn't really give a very definite definition on accessory genes and core genes, because as you say, as the question mentioned, they're like quite a different definition on it. People just usually just use the one frequency threshold to say this is core gene, like above 99% are shared or above 95%. And so wait, do I answer? So I think that I think the wise thing to do is just to use a commonly accepted threshold at frequency to define the core or accessory genes.
Okay, thank you. Yeah, I think maybe the question was also in terms just of, yeah, old terminology. So shared genes would be the core genes,
Yeah,
and
yeah.
then
I mean,
accessory
core
genes
genes
would be
means
the
like
ones that.
most of the isolate carry these genes, so it's shared by most of the isolate. Yeah.
Then the next one is a comment and a question because it's a great presentation. And how does this affect antibiotic resistance in bacteria?
I think it's quite similar to what I talk about on invasiveness. So we know there are like lots of genes that are associated with the antimicrobial resistance, and once these genes, maybe because of vaccination, these genes become rare, then it will quickly increase their frequency. Then you will get more AMR resistant isolates, and that is not a good thing we want.
What percentage of accessory genes comprise a genome?
I mean, the short answer is it's variable between species. And I remember like last time, maybe the conversation between you and me, like also people are interested in human pangenome, and after the percentage in human pangenome will be much more smaller than the bacteria. And also like what I talk about in the asymmetric recombination. So during recombination, it at least in the pneumococcus, it's prefer more deletion than gaining new than insertion. So this will all affect the proportion of the accessory genome in a genome. So there's lots of defense mechanisms in bacteria that will uh, try to inhibit the gaining of new elements in their genome. Mostly because the phage is a continuous threat for bacteria. So yeah, all of these will affect the percentage of the accessory genome in the pan genome.
Can you comment on the role of NFDS in gut microbiota? Broad
I think
question.
yeah.
If
Yeah. Got my yeah. There are some papers about this in like microbiome, and one very direct way to think is it is a very close environment with limited nutrition, and so there are lots of competition between each other. So when I describe NFDS, a lot of scenarios it's all about competition. And when there is a competition, it will maintain everything in a certain frequency. So, and I think maybe the NFDS model, you can easily adapt the NFDS model to this kind of environment.
Thank you. How is the model applicable to organisms of smaller sample size? For instance, for example, for polar bears. So we're moving to a completely different. I think what they are asking is when you work with bacteria or unicellular organisms, you can work with data set with a very big sample size. You can follow very big populations. But how would you work if you have organisms like mammals, where you have very small sample size?
How do we work with small sample size with
Yeah, how would you work with this model? Like, is it possible to apply this model, or do you need like a sample size that is feasible only when you work with unicellular organisms? That's my understanding of the
I
question.
think one important thing is, although I'm talking about an FDS model today, but as I mentioned in my slides, this is extending the neutral model. And if you have, so neutral model is all about randomness. And if we have small sample size, everything can be easily fixed or easily eliminated. So that is a what that is I think that is a major problem we need to think about. So oh yeah, we are talking about an FDS model, but we still need to consider there's a very strong proportion of diversity we observe is from neutral model, and sample size will definitely affect what we observed.
Thank you. So the following one starts with thank you for your wonderful presentation. And then are accessory genes under the NFDS model more likely to occur on plasmids? Is there any particular publication on this that you would like to recommend?
So the figure I showed is actually show what's category enriched in accessory genome. So plasmid and also like the bacterial scenes and mobile genetic elements, those are very enriched in accessory gene. So I think the citation is also there. Yeah.
Okay, perfect. Thank you so much. So the next one is, as an example, there are two strains of bacteria. One is disease-causing and one is not. Based on NFDS, should we expect the disease-causing gene to have more or less accessory genes?
So first of all, I don't think that there's a very definite answer for this, but I think there's several paper talking about the size of more pathogenic bacteria versus the more carriage-prone bacteria. And if I remember correctly, the pathogenic one are smaller in accessory genome size, but I don't think that is a very definite answer.
Thank you. If NFDS can affect by mimicking antibiotic treatments, what information or methods should we have into account for those who work in bacterial resistance field?
Yeah, I think that is
Something
still,
that you should consider.
yeah, that is still a similar question. All I think NFDS is all about predicting like how population maintain the diversity and what is the strain composition maybe after we implement the vaccine. So we can actually know what happened after vaccination. And if we see the fact that we will see lots of AMR resistant strains coming up, then we know this vaccine we might need to consider more. So this is just like and this is just talking about vaccine. We have lots of other with a lot of human intervention. We add antibiotics, and still we can use this kind of concept to predict what happened after we use antibiotics and what is the strain composition after these human interventions. And yeah, at the end, we want the world full of friendly bacteria and without causing lots of disease.
Thank you. Okay. Accessory genes may also present uniquely within an individual or shared among few individuals within a population. In that case, whether neutral model or NFDS would be a better predictor of their frequencies.
Yeah, so yeah, I think that's just the whole point of using NFDS. I think so. NFDS will be, I mean, consider both neutral and NFDS will give you a better description about the population you observe. I think also the important point is if you really have a data set and you want to do some prediction or try to use the model to describe, you still I think it's still wise to do different models and try to compare it. Like using an FDS model is like you adding more parameter, and as most of us know, like adding parameter will cause overfitting. So this is all you need to consider. And also like what I talk about recombination is also add upon on the neutral and NFDS model, you are adding more parameter on recombination. And the thing we need to consider is whether these parameter we add actually give us useful information. Yeah, so